I want to just briefly talk about vision, how the structure of the eye actually does something really incredible, which is it takes something very abstract like light radiation, light waves, and it changes it into a nerve impulse which goes up to the brain. This process is called transduction, and here's how it happens. So we assume that light comes in over here through the cornea and pupil and is focused by the lens. And let's just go ahead and draw that in there. So let's use something dark. So light is coming in like so, and it's hitting the retina like this. And then it has to be transferred. So it's got to go through a nervous system here. So here's kind of a blow up right here, and light is going in this way, and it's hitting the retina. But it's really interesting. What it does is it actually goes all the way through the retina to a basement layer of pigments here. And then it's actually going to go back through the nervous system like this way before it makes contact with the optic nerve. Because remember, look at the optic nerve down here. It's really funny. The optic nerve goes all the way through, and then it makes contact over here, the retina. That's why we have this sort of blind spot here. So once again, the light goes all the way through the retina, then it's absorbed by pigments, and then electrical signals get generated back the other way. Okay, let's take a look. Oh, let me see if I can just drag that picture up here just a little bit so you can see it closer. Okay, so once again, light is going to go through this direction here. There's light. And it goes through all the way over here to a basement membrane here where there are pigments. Now, on top of the basement membrane are two different types of photoreceptors. They are called rods and cones. One way you know the difference is that the cones are bigger. So see this guy here? That's a cone, right? And the rods are kind of thinner. So that's how we're going to tell the difference. And here's their little nucleus here. So they're going to go ahead and do a reaction and change the light into an electrical signal which will then go this way. So these are the photoreceptors, the rods and cones. And then they make connection with a special type of nervous cell here. And these are called bipolar neurons. The reason why they're called bipolar, let's get a different color here, is that there's the cell body here. The cell body actually has two arms that come out of it. So that's why it's called bipolar. And bipolar neurons are very, very rare. This is like one of the few places in the body where you find them. Mostly it's multipolar neurons. Okay, so these guys then take the electrical signal from the rods and cones and they connect them into the optic nerve. Let's get another color here. So now we're in the optic nerve down here, and the cells in the optic nerve are called ganglion cells. There's their little nucleus here. And so these guys are connections of the optic nerve. And then they go ahead and send it to the brain. So light comes in this way. The nerve impulse goes back this way and then goes down the optic nerve. And that's why we have a blind spot. Very confusing, isn't it? Okay, so once again, we can tell the difference between rods and cones because rods are smaller than cones, right? The other thing is that rods are terribly more sensitive. A single light photon is enough for you to set off the rods, whereas cones need much, much brighter light. And that's why under very low light conditions, it's hard for us to tell the difference in color, where we can tell the difference between gray, black, and white, and so forth. Okay, now how do the rods and cones do this? They do something called phototransduction. They are filled with a pigment called rhodopsin. And when rhodopsin absorbs light, it breaks into retinol and opsin. And these chemicals trigger a nerve impulse. And then, after it's finished, they come back together to reform the rhodopsin. So it's a cycle, regeneration. Now, here's the really interesting thing. You may have noticed that if you look at something and you close your eyes, you still kind of see a faded image of it. Now, why is that? I mean, if you're not looking at the image anymore because you've closed your eyes, why do you still see it? And the reason is the chemical reaction that's going on here with rhodopsin is much, much slower than the electrical reaction that's going down the optic nerve. So the signal stops there because you're not getting light, but the reaction of the chemicals is much slower, and it takes a while, and it continues to go on. This is the whole same thing alike, the difference between hormones and the nervous system. Whereas hormones take a while to kind of weed out of your system, you get that adrenaline rush, whereas the nervous system is very, very fast. Okay, finally, I just want to point out one more thing, which will kind of interesting little fun fact here. I mean, humans have incredible eyes, much, much better than almost any animal. But there is a couple of animals that have very interesting eyes. Look at cephalopods like squids or octopus. They actually have a system which has evolved a little better than us, which is kind of strange. So once again, we get light in this direction, right? And then it comes back out this way and then on the optic nerve. Now look at our optic nerve. It comes all the way through the eye, and that's why we have a blind spot there. But look at the octopus eye. Their nerve actually comes and then it stops right here at the bottom of the eye. So they have no blind spot. A little better, although I think given the choice, I'd still rather be a human than a octopus.